Hi there, my name is Dr. Buckley, and it is my pleasure to welcome you to our second lecture video, which provides an overview of the neuroscience of memory. So first, to start, I'm going to review some of the basics. We're going to talk about the structures of the neuron and what action potentials are and how they are transmitted. So the human brain is comprised of billions of neurons, such as the one shown on the slide. So neurons have, in terms of their structure, have a cell body surrounded by branch-like dendrites that receive signals from other neurons. An axon also extends from the cell body and serves to carry signals from the dendrites to the axon terminal. Neurons are not physically connected to one another, but rather they are separated by tiny spaces called synapses or synaptic clefts. Neurons communicate with one another by sending neurotransmitters across these synapses. Making connection across these clefts is crucial for the formation of new memories. Neurotransmitters are stored in synaptic vesicles within the axon terminal. When the action potential reaches the axon terminal, the change in voltage triggers voltage-gated calcium channels that then signal the release of neurotransmitters into the synaptic cleft. The neurotransmitters bind with receptors on neurons in the synaptic gap. If the receptor has an excitatory effect on the postsynaptic neuron, then the neuron is briefly depolarized and an action potential is triggered. If the receptor has an inhibitory effect on the postsynaptic neuron, on the other hand, then the neuron is hyperpolarized, making it less likely that an action potential will be triggered in the future. When two neurons fire at the same time repeatedly, there are chemical changes at the synapse that make the presynaptic neuron more likely to trigger firing in the postsynaptic neuron in the future. This phenomenon is known as long-term potentiation. Some neurons in the areas of the brain involved in memory, such as the hippocampus, have specialized receptors that help ensure that LTP occurs. For example, many hippocampal neurons have AMPA and NMDA receptors, which, when activated, work to build more receptors like themselves nearby. AMPA and NMDA receptors are activated by amino acids of the same name and mediate the synaptic transmission of charged ions. Increasing the number of receptors on a neuron increases the probability that it will fire at the same time as a nearby neuron, which in turn increases the possibility that LTP will occur at the synapse. LTP is essential for memory. When researchers prevent LTP from occurring, memories cannot be formed. One of the most important questions posed by former and contemporary memory scientists alike has been, where exactly are memories stored in the brain? In 1825, future phrenologist Franz Gall proposed that memory, like all human traits, was localized, meaning we have one memory area in the brain. This notion was supported by the work of Paul Broca, who found that damage to a specific area in the left hemisphere resulted in profound agnosia or loss of spoken language, and Carl Wernicke, who found that damage to a different area of the left temporal lobe resulted in profound agrammatism or loss of grammar. The apparent localization of memory for words and rules of grammar suggested that memories were, in fact, localized to specific parts of the brain. In 1921, Richard Seemann, a German zoologist and evolutionary biologist interested in memory, argued in his book The Meme that specific stimuli left permanent memory traces or engrams in the brain. Siemann's hypothesis revived Plato's memory analogy, suggesting that memories were like impressions carved into a wax tablet. In his thesis, Seaman suggested that stimuli produce a permanent record, written or engraved on an irritable substance. Seaman believed that these engrams then instructed the body how to behave, and he used his theory to explain the involuntary coordination between various parts of the body and the brain. Carl Lashley, an eminent American zoologist, set out to confirm the hypothesis that Simone put forth that engrams were localized in specific neurons, and he did this by carefully measuring rat behavior, specifically food-seeking behavior, before selectively lesioning their brains, right? So Lashley argued that if engrams were localized, then a rat should have an engram specifically for food-seeking behavior, and that it would be possible to destroy this engram through lesioning and therefore eliminate a food-seeking behavior. However, Lashley found that no amount of lesioning resulted in the stopping of food-seeking behavior, which suggests the rat did not possess localized engrams in their brain that controlled their behavior. And Lashley instead argues that all parts of the brain contribute equally to the formation of memories and the resultant behavior. So while Lashley is widely praised for disproving Simone's hypothesis that engrams are localized, technically Simone never actually specified whether engrams were located in single neurons or distributed across large groups of neurons. And in fact, uh, Adis and colleagues in 2012 showed that visual areas of the brain are activated during autobiographical memories, as well as more standard areas of the brain, such as the hippocampus. Thus, the current view is that memories are stored in disparate parts of the brain, and memories really are more about connections that are formed between spatially and structurally disparate areas of the brain rather than resulting in a single section. So essentially, the thought, right, is that whenever we retrieve a memory, perhaps of maybe our grandmother making us our favorite cookies, right, that the act of retrieving that memory isn't localized to a grandmother cell or a grandmother neuron, but the activation of that memory of our grandmother making cookies is more likely to um, reside in a disparate and vast array of neural connections, right? So, for example, when we first conjure up a mental image of our grandmother, that activates a region in our visual cortex, and that simultaneously stimulates our auditory cortex when we imagine our grandmother humming a nursery rhyme or something. And then our entire limbic system and hippocampus and amygdala are activated as we feel emotional warmth, if you will. So essentially, the argument that was fundamentally disproven by Lashley was the argument that memories are stored in individual neurons or localized regions of the brain. And contemporary evidence suggests that memories are, rather than being confined to specific neural structures, memories activate really vast networks all throughout the brain. and many different areas of the brain contribute to remembering. So with respect to Lashley's experiments looking at food-seeking behavior in rats, I just wanted to show you this diagram because you'll see in red, in the red lines, all of the areas that Lashley lesioned, right? And lesion essentially just means purposefully damaging an area of the brain by applying an excitatory probe, right? So basically, experimentally damaging the brain and observing the after effects, if you will. So everywhere where you see a line, that is an area that Lashley lesioned. And despite that, the rat continued to engage in food-seeking behavior, which again suggests that memories are much more complex and they must rely on multiple regions of the brain. So, because of Lashley's experiment and many others that have demonstrated that memories are not localized to one region of the brain, but rather distributed across multiple regions and distributed networks in the brain, um, the search for the engram seems to have ended. However, there is some evidence to the contrary. So, for example, Kuroda and colleagues in 2005 conducted a study looking at EEG recordings from single cells. So they looked at patterns of neural firing from single neurons. And so what they did in the study is they flashed pictures of individual people, right? And many of them were celebrities. So for example, they flashed pictures of Halle Berry and Harrison Ford and Steve Carell. And in fact, they found that there were individual neurons that in the temporal lobe that would only fire in response to Steve Carell or Bill Clinton or Halle Berry. So they found preliminary evidence anyway that there are, at least in some cases, individual neurons that correspond to very highly specialized knowledge, such as individual people. So that kind of goes against the conclusion reached by Lashley and others that memories are always distributed across the brain, because in some cases we do have what appears to be a Steve Carell neuron or a Bill Clinton neuron. Now, it's important to consider that there were constraints with this study. So one constraint was the sample of the study. The sample was comprised of individuals who were having surgery to treat epilepsy, right? So one of the treatments for epilepsy is to undergo a procedure where you have specific regions of the cortex removed that are believed to be damaged and causing seizures. So one of the limitations of the study is that because these individuals have epilepsy, it's reasonable to conclude that there might be some brain abnormalities. The other limitation of this particular study is that they only had about 30 minutes to stimulate these individual neurons. So it's possible that if they had had much more time, okay, they could have potentially found other stimuli that activated the Steve Carell neuron or the Halle Berry neuron. But that does seem to suggest that in some cases, at least, memories can be localized, and we do have specific engrams in the brain that correspond to specific memories. Another scientist that I would be remiss to leave out of this conversation is a Canadian neurosurgeon working at McGill University in Canada. So Wilder Penfield was a neurosurgeon who was once deemed the greatest living Canadian, or the greatest Canadian alive, for his work discovering and implementing the Montreal procedure to treat epilepsy. So, one of the hallmarks of epilepsy is that there are typically events that precede seizures. And one of these events that precedes seizures is a sensory experience called an aura. So sometimes an aura can be a sense of smell. So, for example, smelling the scent of lilac or burnt toast. Sometimes it can be some other kind of perceptual sensory experience, such as tasting copper in your mouth. So, having your mouth kind of feel like there's penny in it. Either way, all of these kind of illusory sensory experiences or auras are a very strong indication that a seizure is imminent. So, what Wilder Penfield decided to do, and this was his Montreal procedure, is he would put his patient on the operating table, crack open their skull to expose the cortex, which is essentially the outer layer or outer bark of the brain. And after doing that, after cracking open the skull to expose the brain and the cortex, he would take an electrical probe and stimulate the cortex or the outer surface of the brain. And so his intent in doing that, and by the way, just as a side note or sidebar, this isn't painful. So this wouldn't be painful for the patient because there are no pain receptors on the surface of the brain. And typically patients will get like Versed or some kind of a relaxation drug, right? So Wilder Penfield's intent in stimulating the surface of the brain was essentially to set off an aura, right? So if he stimulated the area of the brain that stimulated an aura, okay, then that would indicate that those cells are damaged and needed to be removed. So, in the Montreal procedure, he would stimulate regions of the cortex until he set off an aura, and then he would excise or remove the damaged neurons. And if you want to see more about Wilder Penfield's Montreal procedure, I did insert a video clip here from a Canadian organization. So if you want to see what it looks like in person, just go ahead and click on that link in the slides, and you can see like a live action portrayal of the Montreal procedure. So a couple of things happened from that Montreal procedure. One is it provided a very promising treatment for epilepsy, and in fact, similar things are being done today by neurologists treating patients with epilepsy, so that's a good thing. The second thing that the Montreal procedure and Wilder Penfield's work led to is something called the sensory homunculus, which is basically a map of the surface of the brain. So on the cortex, Wilder Penfield discovered that different regions of the cortex, different areas of the cortex, correspond to either movements or sensations. So he basically mapped out what we call the motor cortex, if you can see on the slide, and also the somatosensory cortex. So he created a map of the surface of the brain that indicated where the cortical receptors are that correspond to sensation in the gums, in the tongue, in the lips, and the size of the cartoon face there in the sensory homunculus is an indication of how much cortical space or area within the cortex is devoted to sensation or movement of that particular area, right? So the reasons why the lips are so big and the teeth are so big is because we have a lot of sensory receptors on the cortex corresponding to sensation or movements in those areas, okay? So Wilder Penfield discovered the Montreal procedure, treated a bunch of people with epilepsy reasonably successfully, also mapped out the surface of the cortex, and gave us the sensory homunculus. Right. The most important thing, though, for our purposes, is that Wilder Penfield discovered that by stimulating areas of the cortex, not only would people have sensory experiences. So, for example, he would stimulate an area of the cortex and patients would suddenly move their fingers or their arms, right? So that's the motor cortex being stimulated. Or they would suddenly see a shooting star falling toward their nose, right? That would be the somatosensory cortex. But one of the things that Wilder Penfield discovered was that he had some participants who would claim that um, that their memories were being stimulated. So in 1952, Penfield famously reported that stimulating the temporal lobes could lead to vivid recall of specific memories, which led the academic community to believe once again in the concept of localized memories. So, what wasn't noted at the time was that only a small percentage of Penfield's participants, or patients rather, reported any vivid memories when their temporal lobes were stimulated. So it wasn't by any means a generalized phenomenon. Only a few people reported experiencing vivid memories, and those who did often recalled things that could not really be memories, such as seeing oneself standing on the side of the road. So modern critics speculate that those who reported memories during Penfield's stimulation of their brains were more likely reporting dreamlike experiences than actual memories. Thus, Penfield never actually produced good evidence for localized memories, even though many people have been led to believe that he did. So there's still Penfield is certainly still a hero and a legend in terms of all that he gave neuroscience, but in that particular case, the memories that people were reported were more likely dreams, right? So, for example, a patient said something about street corner, and when Penfield asked him where, he replied South Bend, Indiana, on the corner of Jacob and Washington, right? So when asked to explain, he said he seemed to be looking at himself at a younger age, right? Um, so these are the kinds of things that Penfield reported, and he reported that he had several responses of this nature from different patients. He interpreted these reports as memories, and what was striking to Penfield was that they were not the events that people typically recall from their lives, but were rather kind of boring and mundane memories. But the vividness of these reports and their everyday quality led Penfield to suggest that the brain records kind of a stream of consciousness throughout our lifetime, and long-term memory acts like a video recorder. Um, and he theorized that the electrical probe that he applied allowed people to remember and replay otherwise forgotten aspects of their lives. So although this is striking, there are some caveats, right? So first, it is unclear to what extent the reports were actually memories or were experiences generated at the time, right? So these memories may be created in much the same way that dreams are, right? Because the brain does not like randomness, right? It imposes structure naturally on information it's receiving. So to do this, it uses readily available information that is reasonably close to the stimulation. right? So this is information in long-term memory. Penfield's patients were probably getting random stimulation, and their brains were trying to make sense of this random input with whatever knowledge was available in memory, right? So if so, then the reports were kind of mental constructs created at the time from random inputs from the probe, along with information that was stored in memory, right? And that's very much the reconstructive nature of memory. Another problem is that there were very few of these reports. Of the 1,132 total patients that Penfield had, only 3.5% reported these memories. So 24 had auditory experiences like hearing music. 19 had visual experience, so seeing familiar people or objects, and only 12 out of the 1,132 appeared to have complete memory reports. Thus, there's very little evidence to work with. Okay, and some of these reports could not possibly have been re-experiences of past events, right? So, for example, in the report just given, the patient states he can see himself standing on a street corner. If memories of experiences were faithfully recorded, this could not happen, right? You can't look at yourself from a distance without a mirror or a TV camera, for example, right? So it's not really possible for the brain to conjure up a memory in that context. And this is a bit of an aside, but it illustrates how sort of myths of science or misunderstandings get propagated, if you will. Okay, so to continue our overview, following the cognitive revolution, there was a lot of research demonstrating that memory could be short-term, it could be long-term, it could be semantic or episodic, verbal or nonverbal, and that led to the question, to what extent are these systems physiologically and psychologically distinct from each other? So many researchers took to answering this question by looking at memory performance in individuals with brain lesions and by using neuroscientific techniques to look at blood flow patterns in the human brain when a person is completing various memory-related tasks. And the sum of this research, right, the results indicated that different memory tasks do indeed involve different parts of the brain. So, for example, the medial temporal lobe uh, and the hippocampus are needed for encoding new episodic memories or memories for events and experiences in our life. The left temporal lobe is important for verbal memory, whereas the right temporal lobe is important for visual memory. The amygdala, as we've already discussed, is important for encoding memories of an emotional nature, and the cerebellum, premotor cortex, and basal ganglia are involved in learning new motor skills. So the fact that researchers found almost immediately that there were physiological correlates to different types of memory lent credence to the argument that the memory systems were distinct and spurred research in terms of the cognitive neuroscience of memory, which continues to become more and more detailed with advances in technology, which we'll talk about. So continuing with our discussion of the neural correlates, if you will, of different types of memories, we can even look at the different lobes of the brain, right? So the frontal lobe, for example, has a sort of disparate list of functions, right? So a lot of motor actions are coordinated in the frontal lobe. A lot of our attention and processes related to consciousness are coordinated by neural networks in the frontal lobe. Language production, as we've already talked about with Broca's area. Also, short-term and working memory, right? So going back to consciousness, what we experience as consciousness is really just a bundle of neural fibers and neural connections firing in the frontal lobe. And also long-term memory of motor information. So there was a really clever study conducted not that long ago that basically looked at people's ability to memorize different hand shapes, like shadow puppets, basically. And that required a specific area of the brain kind of nestled in the frontal lobe, which is interesting. When we think about the temporal cortex, right, we have the temporal cortex is on the side of the brain near where our ears are. So one really easy way to think about the temporal cortex is it houses the primary auditory cortex, which makes sense for hearing stimuli. It's also responsible. There's Wernicke's area in this region, like we talked about. So language comprehension, or the ability to produce intelligible speech, Wernicke's area is in the temporal lobe. Also, processes related to getting information in and out of memory, so encoding and retrieval processes, and also storing long-term memory representations of sounds and music are found in the temporal lobe, along with obviously the hippocampus, which is a critical structure, and the rest of the limbic system, which is in the temporal cortex. Parietal lobes. So the main function of the parietal lobes is sort of the interconnection of input from all of our senses, right? So that's the primary somatosensory cortex. But interestingly, our long-term memory representations of somatic information. Somatic information is information related to the body, like the body's position in space, and sort of those coordinated perceptions of the body moving in space is found in the parietal cortex. Finally, the occipital lobe is going to be a really critical area, the visual cortex for recognition of visual stimuli, and also long-term memory storage of visual information. So being able to conjure up a memory where you're looking at someone's face, or you can sort of see them in your mind's eye, that's going to be processed in the occipital lobe. So continuing to discuss various structures of the brain and their relationship to memories, we have the medial temporal lobe, which is located deep within the cortex, and many structures that are crucial for memory formation and maintenance are located within the medial temporal lobe, including the hippocampus, the parahippocampal gyrus, and the cingulate gyrus. So we'll talk more about HM, or Henry Molaison, but essentially when his hippocampus was removed, he developed significant anterograde amnesia, which prevented him from being able to store new explicit memories, which we'll talk about. So the hippocampus is part of a network in the brain called the limbic system, located in and below the medial temporal lobe, which is basically just behind your ear in terms of the anatomy. And the hippocampus is considered a subcortical structure. So like most brain structures, it is bilateral. and that is there is one hippocampus on each side or hemisphere of the brain. And to some, its physical shape is reminiscent of a seahorse, hence the name hippocampus, which means seahorse in Greek, right? So the main function of the limbic system seems to involve both memory and emotion, but the hippocampus is a structure very much associated with memory. In particular, the hippocampus is an important part of the circuit that encodes new memories, both conscious and unconscious. It doesn't appear to be involved in the storage of representations of information in memory. However, when we retrieve information, the hippocampus does become activated. Interestingly, the hippocampus is involved in memory across a wide range of species. So rats, monkeys, and songbirds, for example, all have hippocampi that are involved in memory. And the fact that such diverse species use the hippocampus for memory suggests that this brain structure served the function of memory in the common ancestor of mammals and birds many millions of years ago. Along with that, for songbirds rather, the right hippocampus is associated with spatial memory required for migration, for example, whereas the left hippocampus is associated with song memory, which is in this lateralization of the two hippocampi is present in other species as well. So, for example, birds with damage to either the left or right hippocampus become amnesic, right? So, if the left hemisphere is damaged, they can no longer sing. And if the right hippocampus is damaged, they do not fly south properly in the winter, right? So, we see lateralization of the hippocampi across multiple species. In humans, damage to the hippocampus can likewise cause amnesia, and that is memory deficits that are acquired through brain damage. So, in particular, damage to the hippocampus causes difficulties in acquiring new information, right? So, we're shortly going to discuss again the patient HM who had damage to both his left and right hippocampi, and as a result of that, developed a severe form of amnesia that prevented all new learning. And some research suggests that for humans, the left hippocampus takes on more responsibility for verbal memory, whereas the right hippocampus is involved in spatial memory, which is very consistent with what happens in birds as well. Um, and data also show that in humans, damage to the left hippocampus is more likely to affect memory for stories and words, but damage to the right hemisphere will affect memory for directions and pictures, right? And also in humans, damage to the hippocampus can likewise cause amnesia, right? Or memory deficits that are acquired through brain damage, but the way that it presents differs whether we're talking about the left hippocampus or the right hippocampus. The amygdala is also in the limbic system, and amygdala means almond in Greek, again, making reference to the shape, right? And the amygdala appears to play an important role in connecting features of memory with aspects of emotion. It is also highly connected to the hippocampus, consistent with its role in memory, and the hypothalamus, an area of the brain associated with basic emotions, right? So because of these connections, the amygdala is associated with both fear conditioning, which we've talked about, and emotional learning. In humans, the amygdala seems to have an important role in the symptoms of post-traumatic stress disorder, which we'll talk about PTSD and how it affects memory. So when people with post-traumatic stress disorder are asked to retrieve trauma-specific memories, Significant activity can be seen in the amygdala as well as the hippocampus. The diencephalon. So this part of the brain includes structures such as the thalamus and the hypothalamus. So the thalamus in particular is an area of the brain heavily connected to other areas of the brain. It appears to primarily serve as a routing center of sorts, connecting disparate parts of the brain. And parts of the thalamus are crucial in the transmission of information from our sensory organs, such as our eyes and ears, for example, to other areas of the cortex responsible for sensation, right? So it's basically a sensory router. And with respect to memory, the diencephalon includes a lot of different neural connections between the medial temporal lobes and the hippocampus, with the prefrontal lobes, which are involved in memory. So damage to the diencephalon can incur tremendous cost in terms of memory deficits because these areas of the brain are no longer connected. So for example, the amnesic syndrome known as Korsakoff's disease is associated with damage to the diencephalon. Korsakoff's disease involves deficits in new learning, deficits in retrieving well-stored and previously learned information, and an impairment in the ability to distinguish between true and false memories. So as you can see, it's not just in terms of the neural substrates of memory, right? It's not just about the hippocampus or the amygdala or the prefrontal cortex, the prefrontal lobe by itself, but rather the neural connections that connect those disparate parts of the brain and allow them to become activated together. Okay, so now that we've conducted a brief tour of the brain and talked about prominent neuroscientists from the past, now we're going to talk a little bit about how cognitive neuroscientists use various kinds of technology to investigate the cognitive neuroscience of memory. So I'm sure you've heard the specific term a lot. But neuroimaging is the technology that allows scientists to create images that demonstrate which regions of the brain are working during a particular memory or cognitive task. So EEG, or electroencephalogram, is the oldest of the neuroimaging techniques, dating all the way back to the 1940s. So EEG is a technology based on the fact that neurons conduct electricity. This electrical conduction can be measured by sensitive electrodes, which are placed on the skull. And as electrical activity moves from one area of the brain to another, it can be measured as distinct waves of electrical activity, as you can see in the picture. So during particular tasks, some regions of the brain will be more active than others, and this activity will produce a great wave of electricity or produce a larger wave of electricity, which the EEG can detect. More important today is that the electrical activity of the brain can be measured every millisecond, right? Every millisecond, the EEG is taking a measurement. Therefore, EEG is very sensitive to changes in time in the brain. However, when the maximum of 128 electrodes are placed on the skull, EEG is not as good as other neuroimaging techniques at developing maps as to where the processes occur in the brain, right? So another way of saying that is that EEG offers good temporal resolution, but poor spatial resolution. And during sleep, the brain produces characteristic electric waves whose form can be captured on the EEG. And these waves are associated with various stages of sleep, right? And EEG is also important in the diagnosis of epilepsy, right? So to study sleep and epilepsy, the EEG is a wonderful tool. There is also a different kind of EEG known as the intracranial EEG. So with the intracranial EEG, electrodes are placed directly on the surface of the brain. So this can only occur during surgery, right? When the person is anesthetized, right? And we've already seen data from this type of procedure when we talked about the engram, right? So the intracranial EEG is what they used for the Curata et al. 2005 study, right? Where they treated patients with epilepsy. So because recording is now occurring directly on the surface of the brain rather than through the skull and with intervening fluid from the cerebral spinal fluid and stuff like that, the intracranial EEG is much, much more precise in terms of reading the electrical waves in particular areas. So for example, Perez and colleagues in 2015 used an intracranial EEG procedure to follow patterns in the brain when patients were driving car simulators in the hospital, and they found that activity in motor areas of the brain prior to drivers making decisions about Perez found activity in the motor areas of the brain prior to drivers making decisions about routes, suggesting the importance of motor planning in such decision making. So basically, the motor cortex was already activated as measured by these brain waves in the motor cortex, and that suggested that our body kind of acts out motor actions prior to making decisions when it comes to planning out our motor activities. So returning to the scalp EEGs, researchers use a particular method called ERP, or event-related potentials. So in the ERP technique, EEGs are measured in response to particular stimuli or events. So the EEG starts recording when the stimulus is presented to a participant, and it continues for the duration of the trial. The stimulus is then presented in many trials, and the EEGs are averaged across trials to eliminate any random activity that might be present. So what remains is a very clear wave that represents that activity. And once the trials have been averaged together, the resulting data presents a picture of how electrical activity changes over time in response to that stimulus. So for instance, ERP can be used to plot or probe the time course of cognitive processes in the brain. So one example involves a brainwave known as P300. So when words are presented during a memory experiment, a specific wave occurs about 300 milliseconds after the stimulus is presented. It is called the P300 because there's a positive change in electrical voltage. And so in a famous paradigm known as the von Restorff effect, a list of words is presented to a participant. All but one of the words are from the same category, and the out of category word, called the von Restorff item or the oddball, might be the name of a city in California among a list of names of kinds of fish, right? And the P300 is part of the ERP, is distinctly higher for the oddball item than it is for the category items, right? So essentially we can look at the ERP and see exactly where the P300 is and how it correlates to the person's memory. And it allows researchers to make a hypothesis about how memory is processed in the brain. Right? A similar brain imaging technique is a MEG, or a magnetoencephalograph, which allows researchers to measure brain activity by detecting magnetic fields that the brain produces. So there's a lot of physics in this, you guys. So as with the scalp EEG, the measurements take place at the scalp and do not require any invasive procedures. Also, like EEG, MEG can record highly accurate timing of when events occur in the brain down to the millisecond, right? So like EEG, MEG has excellent temporal resolution. And it can also produce more detailed images of spatial maps of the brain. So in this way, MEG is useful in tracking the pathway of information as well as the particular processes that work their way from one part of the brain to the next. MEG presents no risk to the person being studied, and it's so low risk that it's actually been utilized safely with very young infants. So, for example, in one study, Garrido and colleagues in 2012 showed that the amygdala is active when people are evaluating the emotional content of faces. In another study, and colleagues in 2015 showed that specific sensory areas of the brain were active when participants were recognizing words that had been presented to them either auditorily or visually. Right? So there's a wide range of different kinds of cognitive processes that we can examine using magnetoencephalograms, or MEGs. And again, the critical advantage over EEG is that we have both good temporal resolution and good spatial resolution. Positron emission tomography, or PET technology, allows scientists to get a detailed image of a living human brain without having to damage any living tissue. It does involve, however, injecting a small amount of radioactive substance into a person's blood, which does have potentially negative effects. Therefore, it should not be done repeatedly, right? But PET is useful for both medical purposes. So, for example, it can pinpoint a tumor, and research because it can isolate functional areas of the brain. So PET scans offer an enhanced ability to determine where in the brain a particular function is occurring. However, it does not allow for the detailed description of how information is changing over time in the brain. Because it requires about 30 seconds of exposure to capture a good image. Thus, activity in the brain is blurred over a 30-second window. Because of its dangers to patients and the fact that MRI is superior in terms of its spatial resolution, PET scans are seldom used for research anymore, but they're still extensively used in medical contexts. So PET is based on the assumption that areas of the brain that are being used require more blood, and the brain is a biological organ powered by oxygen and sugar supplied by blood. Because neurons that are active require more oxygen, the body should send more blood to those neurons that are engaged in any particular cognitive, emotional, or behavioral task. Therefore, if a researcher can trace where the blood is going during a particular memory or cognitive task, then he or she can correlate that area of the brain with a particular cognitive function, for example. So in PET scans, a small amount of radioactive tracer is injected into the blood of a willing volunteer, and the tracer travels through the bloodstream to all parts of the body and brain. Although all parts of the body receive the tracer, more active areas of the brain draw more blood from our circulatory system than do less active areas. So PET scans use complex measurements to determine which areas of the brain are emitting more radioactivity. So those areas that are more radioactive are associated with whatever cognitive task the volunteer is engaging in. Okay, so I'm sure most of you are familiar with an MRI, right? Um, MRI and functional magnetic resonance or magnetic resonance imaging, as well as functional magnetic resonance imaging, has advanced our understanding of the relationship between the brain and mind more than any other tool. So MRI and fMRI in particular are safe and quick means of generating images of the structure and function of the brain. Moreover, the latest technology involves combining MRI methods to allow detailed spatial mapping, excellent temporal resolution, as well as the ability to look at white matter, which is the axon connections of the brain, and gray matter, which are the neuron cell bodies, right? So MRIs and fMRIs offer much greater spatial resolution of events that take place in the brain than any other neuroimaging technique. fMRI can rescan the brain every 0.2 seconds, thus offering a better time window than PET scans do, although still not as good as EEG. So standard magnetic resonance imaging, or MRI, is a medical tool commonly used to examine structural damage in internal organs and is routinely used to detect anything like a tumor or a growth or other damage in the brain. The term MRI means a structural MRI. So these images are used to produce a detailed picture of the intact human brain. MRI works because different molecules in the brain react differently when placed in an extremely strong magnetic field. To generate structural images of the brain, the detector looks for changes in the structures of water molecules in the brain. An fMRI is a variant of MRI that shows where in the brain particular functional components occur by tracking blood flow. So in addition, the blood flow scan can be superimposed on an MRI to reveal the structure responsible and thereby provide researchers with both a structural map of where of areas of the brain activated during a particular cognitive task, and also a display of the dynamic changes in the brain measured by blood flow. So research using MRI techniques has far-reaching consequences. An example of the power of fMRI to answer previously unanswered questions is one study conducted by Kashino et al., where they were interested in differences in working and short-term memory of faces in individuals with autism. So autism is a disorder in which people have linguistic, social, and emotional difficulties, and particularly difficulties recognizing emotions that are linked to facial expression. So working memory is the memory system that handles information over short periods of time and that we currently have access to. So whatever is currently accessible in our consciousness is indexed in short-term memory. And it turns out that individuals with autism have a deficit in remembering faces, and Kashino and colleagues wanted to determine whether it was a perceptual phenomenon or a memory phenomenon. So if it was a perceptual phenomenon, the individuals with autism would have difficulty seeing the faces, and this difficulty would show up in the fMRI as decreased activity in the areas associated with vision. So for example, the visual cortex. If it is a memory phenomenon, then individuals with autism would see the face, but then have difficulty matching it later to representations in the brain, right? So this would show up as an fMRI decrease and as a decrease in activation in memory areas such as the prefrontal cortex. So Kashino et al. Asked their participants with and without autism to match previously learned faces while being monitored in the fMRI. And the researchers found that relative to individuals without autism, individuals with autism showed lower levels of activation in the areas of the left prefrontal lobe, known to be involved in working memory. Thus, the neuroimaging data support the memory interpretation of this deficit in autism. Another fairly new MRI technique is diffusion MRI or diffusion tensor imaging or DTI. And DTI compares patterns of the movement of molecules, particularly water, within tissue of the brain in order to create structural images. And DTI is particularly helpful for examining white matter connections in the brain. So it's particularly useful for delineating pathways in the brain that connect neural centers rather than looking at neural centers themselves. So for example, Dick and colleagues in 2014 looked at the white matter pathways that connect areas of the brain associated with language. These pathways, such as the arcuate facialis, are particularly involved in these processes. DTI is also very helpful related to memory in the sense that it's helpful in diagnosing Alzheimer's as opposed to other forms of dementia. So if someone is experiencing memory difficulties, diffusion tensor imaging would help to delineate which pathways in the brain are no longer firing properly, essentially. So diffusion tensor imaging, or DTI, is an MRI technique that compares patterns of movement of molecules within tissue to create structural images like the one on the slide. And next we have brain stimulation techniques. So another class of neuroscience techniques involve directly stimulating specific regions of the brain and then observing the change in cognition that results after the stimulation. So there are several of these brain stimulation techniques, and we'll talk about each of them. So we have transcranial magnetic stimulation, transcranial direct current stimulation, and deep brain stimulation. So in transcranial magnetic stimulation, or TMS, a magnetic charge is applied via the skull to a particular area of the brain, and this is done by placing a magnetic coil near the surface of the scalp. This coil then produces an electric current or electric currents, which surge into the adjacent areas of the skull and brain. So TMS has a number of medical uses, including being an effective treatment for severe migraines. In terms of research, TMS allows a small pulse of electricity to temporarily disrupt function in a small area of the brain. So researchers can then observe the changes in cognition and behavior that follow. So in some cases, TMS will inhibit performance or decrease people's ability to do a task, but in some other cases, it can also enhance skills. In either case, the effects of the TMS wear off within a matter of minutes after the coil is removed from near the person's scalp. So because TMS allows true experiments, one can infer causation from the disrupted area rather than simply correlation as in neuroimaging work. Right? So some examples of TMS research on memory include a study by Desmond and colleagues in 2005 that showed that verbal working memory, our working memory for words, was impaired after a single pulse of TMS was transmitted to an area of the brain called the cerebellum. In addition, Bonney and colleagues applied a continuous TMS to an area of the brain known as the precuneus. And stimulation of the precuneus led to a better ability to recognize the context of a particular memory. That is, participants were better able to recognize previously seen pictures and identify the color of these pictures. In transcranial direct current stimulation, or TDCS, low current electricity is applied directly to the scalp in a continuous fashion. Right? So in TMS, the coil is kept away from the scalp, and the current can either be a single pulse or continuous, but in TDCS, the coil is in constant contact with the scalp, and the current is always continuous. So this technique has a number of really critical medical applications, including improving cognitive performance after a stroke, alleviating depression, and also reducing memory deficits in early Alzheimer's disease. Go. And it's used for both research and medical reasons. So as far as research goes, TDCS can help localize function in the brain. If current is applied to an area of the brain and performance on a certain task improves or gets worse, that area of the brain must be involved in that cognitive skill. So for example, Schall and colleagues in 2015 did an interesting study using TDCS, looking at the role of the right parietal cortex in the recognition of recently learned melodies. So melody refers to the main line of a song, right, or the musical accompaniment. And we generally hum when we think of a melody, right? So in the study, participants listened to novel melodies that they had never heard before, derived from folk songs. And after the music had been listened to, the TDCS was applied directly to the scalp above the right parietal cortex. In a second experiment, the TDCS was also applied to the left parietal cortex to serve as a control. In addition, there was a group of participants who were set up with all the TDCS equipment, but the current was never turned on. So this also served as a control, or sham condition as it's called. And when the TDCS was being applied to the right parietal cortex, participants recognized fewer of the melodies as old, okay, or as songs they previously heard, than did participants in either control condition. Thus, stimulation of the right parietal cortex interferes with memory for musical melodies. Because of this interference, one can speculate that the right parietal cortex must be an area critical in music perception or musical learning. In deep brain stimulation, or DBS, a device is implanted directly into the brain, which then sends electrical impulses to specific regions of the brain. So DBS is exclusively implanted for medical reasons. So it's useful for alleviating, for example, symptoms of Parkinson's disease and other brain-based motor disorders. It has also been successful in treating major affective disorders, such as obsessive compulsive disorder, and also major depressive disorder. In research, it is only used with patients who have the DBS device implanted for medical reasons. However, in a population of severely epileptic patients, Miller and colleagues in 2015 found that DBS pulses to the fornix area of the brain improved the patient's performance on a variety of memory tasks. And this also makes sense because DBS not only alleviates the motor symptoms associated with Parkinson's, but it also improves cognitive functioning as well. So the oldest methodology for examining the relationship between memory and the brain, as I've already mentioned, is to study patients who have experienced some kind of damage to the brain. This is because examining patients with neuropsychological deficits does not require any kind of sophisticated technology. However, researchers must first locate patients who have suffered brain damage and then observe the cognitive and behavioral deficits in the patients. So going back to the famous case of Phineas Gage in 1848, right? Research has been directed at how brain damage affects cognition and behavior, right? So Gage was a construction foreman on a railroad crew who was severely damaged when a poorly timed dynamite blast shot a metal rod directly through his frontal lobe. And although he survived the accident and lived for many years afterward, some of the accounts of his brain injury suggested that he had fairly pervasive impairments in terms of his cognitive and emotional processing faculties and also his personality. So this study of the change of behavior kind of set the stage for the development of neuropsychology. And the goal of neuropsychology is to correlate behavioral deficits or cognitive changes with the area of the brain that has sustained damage, right? So the assumption then is that the damaged area of the brain normally involved in the function of the affected behavior or cognitive ability is now altered. And because damage to the area causes deficits in a particular function, such as working memory, for example, then it is thought that the brain area must play some role in that function. So just over 100 years after Phineas Gage had his accident, in September of 1953, a 27-year-old man, Henry Molaison, underwent a very risky experimental surgery to alleviate symptoms of his debilitating epilepsy. And during the surgery, parts of his medial temporal lobe, including most of his hippocampi, were removed on both sides. So as a direct result of the surgical procedure performed by Dr. Scoville, Henry Molaison suffered significant anterograde amnesia, which is a deficit in learning and retaining new information. He could not learn new facts, such as memorizing a phone number. He also suffered some relatively mild retrograde amnesia. So, for example, his memory for some past events before the injury became less accessible. And although this surgery has never been repeated on any other human being, Henry Molaison's memory was studied exclusively for the next 50 years. So Henry passed away in 2008 at the age of 82. And although his ability to encode new events into his episodic memory was strongly affected, research shows that his short-term memory or working memory and procedural or implicit memory were largely intact, right? So many other patients have been studied since then, and these patients have varied from having very mild amnesia with memories just barely different from those of people without brain damage to very severe amnesia. Moreover, the particular pattern of deficits is different in each patient, and the pattern of these deficits can be linked to where in the brain the damage occurs in that patient. Um, so, neuropsychological studies allow researchers to examine the relationship between deficits in cognition and behavior with the locus or area of damage within the person's brain. So, in fact, most brain damage is fairly diffuse and spread around large areas of the brain. However, in some cases, often the result of bullet wounds or strokes, and sometimes surgery, the damage can be quite localized, allowing clear correlations to be drawn between the memory deficits they experience and the particular site of the brain damage. So just a little bit more background on Henry Molaison, who is known in the literature as HM. So Henry Molaison had a fairly unremarkable life, and by that I mean just a normal life, up until he was about the age of 6, and he fell out of a tree as a child and sustained a significant blow to the back of the head, which resulted in very, very profound, intractable epilepsy. He was having anywhere from 30 to 50 seizures a day. And at that point, neuroscience was in its infancy, so there wasn't any sort of medication. Like nowadays, we have a lot of benzodiazepines, anticonvulsants, and other drugs that can manage epilepsy fairly well. But in the 1950s, there simply wasn't those kinds of treatment options available. And so essentially what happens with epilepsy is you have a totally uncontrolled electrical storm in the brain caused by far too many excitatory signals and not enough inhibitory signals being fired by various action potentials. And so one of the neurotransmitters in the brain that gets excessively transmitted during seizures is calcium. And one of the structures of the brain that causes a lot of that excitatory activity is the hippocampus. And so previous surgeries with epileptics, they had taken just a section of the hippocampus away because the predominant excitatory neurotransmitter in the brain, glutamate, is produced by the hippocampus. And so just removing a section of the hippocampus was adequate for controlling a lot of people with seizure disorders. But given how extreme Henry Molaison's case was, Dr. Scoville, the neurosurgeon that performed the procedure, decided to remove the entire structure. And because of that, Henry developed profound anterograde amnesia. And so what we learned from Henry Molaison was essentially the dissociation between short-term and long-term memory, and also the dissociation between implicit and explicit memory. So short-term memory is, again, whatever your consciousness is currently focusing on, right? So HM, or Henry, could very reliably, if you told him to, count to 10 or to rehearse a phone number, he could do that indefinitely as long as he didn't stop rehearsing it, right? So he could hold things in consciousness, in short-term memory, but because his hippocampus was destroyed, he could no longer convert those transient short-term memories into long-term memories. So that was the first association. And the second association was between implicit and explicit memories. So believe it or not, HM was perfectly able to do tasks that relied on the neural circuit for implicit memory. So his hippocampus was destroyed, which meant he could no longer form new explicit memories. So he couldn't remember any of the experiences or knowledge he had access to after the surgery. But um, HM could be taught to classically condition things. And they also gave him the many researchers that interacted with him, also gave him a mirror drawing task. And so with a mirror drawing task, you basically draw a picture relying exclusively on a mirror, which is a very, very difficult motor task. Um, uh, and it takes many, many practice trials for anyone to become proficient at this. And so they gave HM this mirror drawing task, and they had him do it over several sessions. And interestingly, even though he had no memory of those specific researchers and he had no memory of that particular task, his performance did increase, so much so that he was able to seemingly effortlessly draw this picture in the mirror, and he regarded it as being a very easy task, even though in actuality it was very difficult because he had no memory of performing those earlier practices. Another thing I just want to mention, because this is honestly one of the most tragic cases that any memory scientist, is aware of. It really pains and aggrieves a lot of people, particularly me, knowing that his life was ultimately destroyed, and that he had to live in an institution for the rest of his life, and basically from the age of 27 until he died at the age of 82, he lived kind of in isolation. And one of the most heartbreaking components of this case is just that he was such a nice guy. He was just the sweetest man. And unfortunately, I never got a chance to meet him, but my graduate advisor, Robert Bjork, and a few other people that I do research with, can recall meeting him and just commenting on what a lovely, lovely man that he was. And one of the things that I heard from someone that actually got to meet Henry several times is that when they were initially doing this testing on him, the mirror drawing and the other memory tests that they did, they would basically take him from the hospital where he was to a testing facility about 45 minutes away. And so they would drive him up to this facility, and one time they were in the car driving up to the facility, and someone had gone to McDonald's and gotten a drink of some sort. So there was a McDonald's cup in the console of the vehicle. And apparently Henry's first job was working at McDonald's. And so he told this really funny story about his first job at McDonald's, and he said it with a lot of emotion, a lot of laughter, a lot of charisma. But unfortunately, because of his amnesia, he ended up telling this story probably 30 so times over the course of this 45-minute period. So that example sort of very, very clearly indicates really how densely amnesic he was. The other thing I wanted to mention is that there is a book. There is a book titled Patient HM that was written by his great grandson. And unfortunately, it is not really a very accurate portrayal of what happened. And there have been many, many memory scientists and people that have basically come out and said that the book is not accurate in terms of its portrayal of HM at all. So I just wanted to warn you, if you're interested in HM, I would be happy to supply, books and papers and pictures, and anything else. But just to say that that patient HM book is not very accurate at all. So I can definitely recommend other resources if you would like to learn more about Henry. So now that we've talked about various techniques that cognitive neuroscientists and cognitive psychologists utilize to study memory, I wanted to talk a little bit about how different drugs affect memory. So firstly, we can talk about drugs that essentially induce a state of amnesia. So believe it or not, benzodiazepines are the most frequently prescribed drugs in the United States. And these are drugs such as Valium or Ativan or Triazolam or Midazolam. And they're usually prescribed to alleviate everything from anxiety to insomnia to muscular spasticity and other things. And one of the significant downsides of these drugs is that while they treat those conditions very effectively, they're also very bad for memory. So they induce very significant symptoms of amnesia, and they impair both episodic and semantic memory. So this is critically important in a lot of criminal cases where you have individuals who were under the influence of some of these drugs and then committed crimes, but virtually have no memory of having done the crime. And this is particularly compounded when you have somebody that's using both benzos and alcohol, because alcohol is also a central nervous system depressant, and can cause a lot of amnesic effects. So it becomes problematic, for example, if you're trying to read somebody their Miranda rights and they don't really have a memory of the previous act or really what's going on at all. So another drug that has been studied extensively with respect to its effect on memory is caffeine, right? So unfortunately, there's no clear delineation at this point. The data is fairly mixed. So some experiments or some articles suggest that caffeine can be helpful for memory, and other data suggest that it's actually not helpful at all. And so basically, the issue comes down to the fact that caffeine might not promote actual efficiency in learning, but rather it might allow you to stay awake longer, which is actually not helpful in terms of your brain's ability to acquire information. So it's helpful for memory if you need more time to learn something, right? But the time you spend learning it while on caffeine, right, is not going to be as efficient as if you weren't under the influence of caffeine. So whereas you might be able to learn 20 new units of information when not caffeinated, you're able to stay awake for the full hour or whatever unit of time, but you might only be able to learn like 18 or 15 units of information while under the influence of caffeine, right? So really, if you need to stay awake, okay, caffeine is helpful. If you need more time to finish a task, caffeine is helpful. But if you want more bang for your buck in terms of what you're actually going to be capable of learning during that time, it's best to not be under the influence of caffeine. So now let's talk a little bit about things that don't work. So for a long time, people have drank like tea with leaves from the ginkgo tree, thinking that it was helpful as a memory enhancer. However, researchers have looked extensively at this through meta-analyses and other things, and there's virtually no data that suggests that this particular herb is helpful for memory. So it might taste good and be refreshing in a tea, but it's not actually going to help you remember anything. Okay, so another example, like the leaves of the ginkgo tree, of a drug that does not affect memory or improve memory rather, is the drug Prevagen. And I do recognize that this is a personal like axe to grind on my part. But I don't know if you all who are watching this live in Bibb County in Macon, and you've noticed this, but the commercials for Prevagen are specifically targeted toward like senior citizens and people who are dealing with age-related memory failure. But basically, there's no clinical evidence that it actually improves memory. So what is Prevagen? Prevagen is an over-the-counter dietary supplement that claims to improve memory and brain function, especially for older people with mild memory loss. But when we actually look, okay, at what the active ingredients are in this drug, there's vitamin D3, which is essentially fortified calcium, and there's also a protein found in jellyfish. Okay? Does that seem like a magical cure-all? Probably not, right? And so the biggest problem with Prevagen is that they're claiming that their active ingredients support memory retrieval in older adults. But the very nature of the supplement, okay, is essentially the active ingredients in this supplement get absorbed during digestion. Right? One of the biggest reasons why these claims are suspect is our one significant issue is that the protein found in Prevagen is likely broken down in the digestive system before it can reach the brain. So there's no substantial evidence that this protein can even survive the digestion process long enough to be absorbed by the bloodstream, cross the blood-brain barrier, and then perform its intended function, right? So if our stomach is breaking down the supplement before it even gets to the brain, that calls into question all of those claims that it supports memory, right? Also, none of the studies that support Prevagen's effectiveness are independently evaluated, and most of them have been thoroughly discredited because they're paid for by Quincy Bioscience, which is the company that makes Prevagen, and they lack appropriate experimental controls and other features of proper experimentation. And in fact, when we hold them to scrutiny based on basic scientific standards, there's no evidence that Prevagen improves memory. So in addition to that, the U.S. Federal Trade Commission and the New York Attorney General have filed complaints against Quincy Bioscience, alleging that the company made false claims about Prevagen's ability to improve memory. And the FTC and other experts argue that the company's studies do not provide substantial evidence to support the claims they make. Even the studies that they basically paid for did not provide adequate consensus. And so the broader scientific community remains skeptical, and the consensus is there's no solid evidence to support the idea that taking this dietary supplement actually improves your memory in any way. So a lot of the observations of people that claim to have seen benefits, the placebo effect can't be ruled out, which is essentially perceived improvements simply because they believe they are taking something beneficial, right? But why is this problematic? Well, it's problematic because if you're an older person who thinks that taking Prevagen is going to help you, okay, then you might not seek out a doctor's care. You might not go see a neuropsychologist who can actually conduct evaluations for dementia and Alzheimer's. And more importantly, a lot of older adults are living on Social Security and have very limited income. So the fact that they would spend money on a supplement that essentially does nothing for them is just problematic from an ethical standpoint. One last thing I wanted to mention before I end this video is the interesting relationship between olfaction, or our sense of smell, memory, and the brain. So olfaction is our sense of smell. And human beings have long been aware of the intimate relation between the sense of smell and memory, particularly the retrieval of highly personal autobiographical memory. So most people can describe the relation of a particular smell and to some salient event from their life, right? And in fact, they've done studies where they ask people to do this. So for example, maybe the smell of mothballs always reminds you of your grandmother's house, or the famous writer Marcel Proust, for example, described how the scent of a French pastry called a madeleine mentally transported him back to his childhood in the south of France, right? So many people report associations between a particular scent or perfume, like cologne, with a particular maybe boyfriend or girlfriend or a relationship that you've had from years ago, right? So I had a friend who associated the smell of Pantene Pro V shampoo with Christmas, because her mother always bought it. It was like an expensive shampoo, so her mom always bought it during the holidays, right? So as is clear from these examples, the connection between memory and smell also has ties to emotion, right? So the memories elicited by odor are usually emotional in nature. And the neural reason for this is the strong connection between our sense of smell, emotion, and memories. And this is because all of the brain structures involved in these three processes make up what's called the limbic system. So the limbic system is involved in both memory and emotion, but it's also the primary area for processing odors. So located within the limbic system is the olfactory bulb, the primary organ in the brain for processing odors. It receives input directly from olfactory nerves coming from hair cells in the nose, kind of gross. And only after information passes through the olfactory bulb does it go to higher areas of the brain and the cortex. But the olfactory bulb is heavily connected, neuronally speaking, to important memory systems in the limbic system, most notably the hippocampus and the amygdala. And these strong neural connections provide the basis for the strong association between odors and both memory and emotion. Right? Interestingly, it is only after these connections between the olfactory bulb and the limbic system occur that information is processed by the olfactory cortex and other areas in the prefrontal lobe. So this may account for the gut feeling that is characteristic of these strong odor memory emotion associations, right? So again, The olfactory bulb is the primary organ for processing in the brain for processing odors. The limbic system is involved in both memory and emotion, but it's also an area of the brain responsible for the processing of these odors. And so the olfactory bulb connects to both the hippocampus and the amygdala, which explains the association between odors and strong emotional autobiographical memories. Okay. Thank you so much for your attention. I hope you enjoyed this video, and I hope you have a wonderful day.